We can see the DNA letters, but we cannot directly see where the gene is. The HMM tries to uncover the hidden gene structure. Each region of the genome has its own DNA pattern. Exons may look different from introns. The robot does not jump randomly. It follows biological rules. Start codon before exon, intron between exons, stop codon at the end. Let's make our HMM more biological. The DNA sequence is CCC8EG-CGCTA. Biologically, this represents an intergenic region, a start codon, an exon codon, and a stop codon. To model this accurately, we split the gene into smaller states. The start codon must follow A, then T, then G. The stop codon must follow T, then A, then A. So the hidden states move step by step through the gene structure. Intergenic, start, exon, stop after gene. This allows the HMM to follow real biological rules instead of making random state jumps. Now we turn the biological idea into code. We list every hidden state step by step. Step 1. Define the hidden states. We begin by defining the biological states: intergenic, start codon, exon codons, stop codon, and after gene. Step 2. Load the DNA sequence. Next, we provide the DNA sequence, CCC8EG-CGCTA. Step 3. Set the starting state. The model starts in the intergenic region with 100% probability. Step 4. Define biological transitions. We specify how the model can move between states, following the biological path from intergenic to start, through exons, then to stop, and finally after gene. Step 5. Define DNA emission rules. Finally, we tell each state which DNA letters it can emit. The start states produce 8G. The stop states produce TA, and the exon states emit typical coding region bases. Now, let's implement the Viterbi algorithm. First, we initialize the probability of starting in each hidden state using the first DNA base. Next, we move through the sequence one position at a time. For every current state, we test all possible previous states. We multiply the previous probability, the transition probability, and the emission probability. We keep only the most probable path leading to each state. This process repeats until the entire DNA sequence has been analyzed. Finally, we choose the state with the highest probability at the last position. The algorithm returns both the probability of the best path and the most likely hidden state sequence. Next, we give the DNA sequence to Viterbi. The algorithm follows the biological rules encoded in the HMM and predicts the most likely gene structure. The HMM successfully identified the start codon, coding region, and stop codon. It has converted raw DNA into a biological gene model.